Hello everyone. Now today, before I begin with today's topic, I simply want to ask you all that have you revised the reproductive systems that we have done earlier, the male reproductive system as well as the female reproductive system, because today what I am going to teach you, that is gametogenesis, is related to the structure of the primary sex organs, testes as well as ovaries. So let's begin today's class. Gametogenesis, as I've already written the topic. If you remember, I told you genesis means formation. So formation of gametes, and we know gametes are formed in the primary sex organs. So in males, testes is the primary sex organ, and formation of gametes, that is the sperms, by the testes is called spermatogenesis. The term itself is telling you genesis means formation of sperms. So in male the process is called spermatogenesis. Same way in females the primary organ is ovary, pair of ovaries, and the process is called oogenesis, genesis of ova. So this is basics about gametogenesis. Now one by one we will be dealing with each process. First comes spermatogenesis. Now I said you have to have an idea of the primary sex organ. If you remember, I told you testes is made up of seminiferous tubules. We had done the TS of testes. Suppose this is a seminiferous tubule. These seminiferous tubules are lined with germ cells, and in between there were Sertoli cells. So actually these germ cells are going to form the sperms. Now coming to this, the sperm cells are formed from these germ cells. So these germ cells are called spermatogonia. They undergo 3 stages to finally form the sperms. Sperms are filled, suppose this is the lumen of the seminiferous tubule. You remember I told you sperms are produced and they collect in the lumen of the seminiferous tubule. Spermatogonia are formed. This spermatogonia finally leads to the formation of the sperms, and there are 3 stages that take place. First stage is called the multiplication phase. Second phase is called the growth phase. And the third phase is called maturation phase. Now these spermatogonia undergo several divisions, and all these divisions are mitotic in nature, and in mitosis you have already learned mitosis and meiosis in detail in class 11. So from one cell 2 are formed. These divide again. Each cell forms 2 cells. So that's how the number goes on increasing, and that is why we call it multiplication phase. But mitotic means the chromosome number remains same. Spermatogonia is a diploid cell having 2n number of chromosomes, which means it has 46 chromosomes. And when it is dividing mitotically, all these in the multiplication phase will also have 2n number of chromosomes. So out of these, one of them is going to mature. One of them will grow and mature, and it will be called the primary spermatocyte. This primary spermatocyte then undergoes meiosis. Meiosis, you have learned, takes place in 2 phases. First phase is called meiosis 1, where actually the chromosome number is reduced to half. So in the first meiosis, which is actually a reduction division, the chromosome number, which was 2n, has now changed to n. That is, it becomes half. These two. Then it is called secondary spermatocyte. So this was the multiplication phase where several spermatogonia are formed. One of them matures, grows, and is called the primary spermatocyte. Then meiosis takes place. This comes under the third phase, that is the maturation phase. Meiosis 1 takes place. Secondary spermatocytes are formed. These secondary spermatocytes then divide mitotically. The second phase of meiosis is actually a mitotic division. So here the chromosome number has already become half. From 46, it has become 23. Both these cells have 23 each. So now in the second meiotic division, more cells are formed, each having n number of chromosomes. That is here, see, from n. That is why it is called, this is a kind of mitotic division. The second meiotic division is a kind of mitotic division because now the chromosome number remains same. Now at this stage, we call them spermatid. So you see, from spermatogonia, primary spermatocyte in the growth phase, then secondary spermatocyte, and then we have spermatid. This stage, spermatid, the spermatids that are formed, they attach to the Sertoli cells. Sertoli cells, if you remember, I told you, provide nutrition. So basically, these spermatids attach to the Sertoli cells for three main reasons. They get support, they get protection, and at the same time, they get nutrition. And a major change takes place over here when they are attached to the Sertoli cells. The spermatids are attached to the Sertoli cells and a major change takes place. A major change leading to maturation and formation of sperms. That is called spermiogenesis. Because now the sperms are going to be formed. So what is that major change? Each of these spermatids lose a little bit of cytoplasm. The nucleus gets condensed and a big head is formed. Head of the sperm is formed and a flagellated tail is formed when it is attached to the Sertoli cells. Head and tail. Head and tail. Like this. And this is then released from the Sertoli cells and they collect in the lumen. So these are the sperms that have collected. Sperms are also called spermatozoa. So these are the sperms that have collected in the lumen of the seminiferous tubule. I had shown you the structure of, just to explain if you did not revise what I taught you earlier. So with this it will become clear that seminiferous tubules form the testes and sperms are developing in the seminiferous tubules, and a seminiferous tubule has germ cells and Sertoli cells. So the function of both, actually the germ cells are forming the sperms. So this release of sperms after spermiogenesis from the Sertoli cells into the lumen is called spermiation. So before I erase this, now you see what are the different terms. Spermatogenesis is formation of sperms in the seminiferous tubules. This is a structure of one seminiferous tubule. S seminiferous, T tubule. And the three phases of spermatogenesis is number 1 multiplication phase, number 2 growth phase, number 3 maturation phase. Multiplication means several spermatogonia are formed. Each of them are diploid. One of them is going to grow up and form a primary spermatocyte. So this comes under growth phase. This is growth phase. This is multiplication phase. Then after that, the third phase is maturation phase, where the primary spermatocyte matures to form the secondary spermatocyte after the first meiotic division or first reduction division, forming two haploid cells now. These haploid cells then undergo the second meiotic division, which is mitotic in nature, forming four haploid cells. These four haploid cells are called spermatids, which will attach to the Sertoli cells for maturation, getting nutrition, protection and support from the Sertoli cells, and the process is called spermiogenesis. These two terms are very important because the board often asks questions on these two terms. You have to define and explain these terms. So spermiogenesis is maturation of the spermatids forming sperms or spermatozoa after getting nutrition, support and protection from the Sertoli cells. Now after this they are formed, their release into the lumen of the seminiferous tubule is called spermiation. And structure of the sperm we have already discussed, but if you have not studied, we will quickly revise. It has a spoon-shaped head with a large condensed nucleus. The tip is acrosome. Then we have a neck having a pair of centrioles, a middle piece having lots of mitochondria arranged spirally, and a flagellated tail. This portion is called the tail. This portion is called the head. Tail is divided into four parts: a neck, a mid piece, a neck, this is the principal piece, this is the end piece. So this was the structure of the sperm. So we have done spermatogenesis in detail now. So I'm going to erase the whole thing and then we will do oogenesis today. If you want to note. Okay. Now coming to oogenesis. As the term tells you, oogenesis is formation of ovum. Oogenesis is formation of ovum, and for this we will be needing the diagram of the structure of the ovary. But before I draw that, oogenesis also takes place in three main steps or three main phases. What are these? The first is multiplication phase. Second is growth phase. And the third is maturation phase. Basic steps of gametogenesis are the same in spermatogenesis and oogenesis. Now coming to the ovary. If you remember, ovary was an almond-shaped structure like this, attached to the body wall by mesovarium, and this was lined by the germinal epithelium. So this germinal epithelium produces several cells. These are called oogonia. So oogonia undergoes several rounds of cell division, which we call multiplication. So suppose this is the multiplication phase. Each of these has 2n number of chromosomes, that is 46 chromosomes. Then in the growth phase, one of them is going to mature. One of them is going to mature to form the primary oocyte, and the others will encircle it and surround it to form follicles. That is why we use the term as primary follicles. What are follicles? Follicles are actually collection of oogonia. One matures and grows in the growth phase to form the primary oocyte, and the others encircle it, and then these are formed at the time of birth, and after that they are not going to increase in number. Slowly they will degenerate. So at the time of puberty, these are limited to 60,000 to 80,000. That is why a female is not fertile throughout her lifetime. Because a stage comes when these follicles finish and production of ovum by the female stops. When we do the menstrual cycle, I will explain this to you. So this is 60,000 to 80,000 follicles are there at the time of puberty, and these are going to last for the whole say the reproductive phase or sexual cycle in a female. So this primary oocyte encircled by the other oogonial cells, oogonia, are the primary follicles. These then form, a cavity is formed, and we say that the follicles are maturing, and finally the follicle that matures has a cavity. Now I will show this structure as in labs. This is the secondary oocyte surrounded by other cells, and it has a cavity. This cavity is called antrum. It is filled with a fluid. This is the oocyte, the primary oocyte, and this is zona pellucida. This zona pellucida is formed by the primary oocyte and the other cells that were surrounding it. The other cells that were surrounding it in a follicle, that is the follicular cells and the oocyte together form this. So this is a structure of a mature follicle, and we call it a Graafian follicle. Now this primary oocyte undergoes, till here the chromosome number is diploid. It has 46 chromosomes in it. This then undergoes the first meiotic division. First meiotic division to form two cells. Now these two cells, one is important, which will be the secondary oocyte, and the other is called a polar body. Chromosome number has reduced to half after the first meiotic division, but only one oocyte, secondary oocyte, is formed. The other one is called a polar body. So at this stage, this is secondary oocyte in a mature follicle, Graafian follicle. This is the secondary oocyte. At this stage, the follicle ruptures and releases the secondary oocyte in the peritoneal space between the ovary and the oviduct. In a female reproductive system, in my last class, I had told you. This is then picked up by the oviducal funnel or the fimbria of the infundibulum. So at this stage, when it moves up to the uterus, it is still in the form of secondary oocyte. Only at the time of fertilization, this secondary oocyte will form the ovum, and another polar body. Another polar body will be formed. This is second meiotic division, which is mitosis. Actually, is mitosis because chromosome number remains same. This is second meiotic division, which is mitotic in nature. And this is formed at the time of fertilization. The polar body forms two more polar bodies. So finally at the end of oogenesis, we have only one ovum and we have three polar bodies. So this is a mature Graafian follicle. And now after it has released the secondary oocyte, it changes into the corpus luteum. And corpus luteum will exist in the ovary till fertilization occurs. If fertilization occurs, Graafian follicle, which has changed into corpus luteum, will persist throughout the period of gestation, throughout the period of pregnancy, and will help to maintain the pregnancy by producing hormones. If fertilization does not occur, then the Graafian follicle degenerates by the 28th day of the ovarian cycle. So this was oogenesis. Now another important thing that I just pointed out, that these oogonia are limited. They are 60,000 to 80,000 at the time of puberty, and each ovary is capable of producing one ovum in a month. So alternately, once ovum is produced from one ovary, then the ovum is produced from the other ovary. So this is oogenesis. Again, for a quick recap, 3 phases in the formation of the ovum: multiplication phase, where the oogonia multiplies, then the growth phase, where one grows big and forms the primary oocyte, then comes the maturation phase, where the first meiotic division takes place and chromosome number becomes half. One secondary oocyte is formed and one polar body is formed. Then this secondary oocyte is released from the ovary and is picked up by the oviduct and moves up in the oviduct to the uterus, and if it comes in contact with the sperms, fertilization occurs and the ovum is formed at that time. Finally, 3 polar bodies and 1 ovum is formed. This is the structure of the Graafian follicle. Now, the structure of the ovum. The ovum is surrounded by perivitelline space, by zona pellucida, and lot of follicle cells. This is the ovum. This space is called perivitelline space. This is zona pellucida, which is formed from the by the primary zona pellucida, which is formed by the primary oocyte and the other follicle cells that were surrounding the primary oocyte, and this is called corona radiata. And all this is there in the granular cells, and a cavity called antrum is there. Only at the time of fertilization this ovum will be released. The sperms will penetrate through these layers and finally the fusion will take place. This I will explain when I do fertilization in detail. So this was about gametogenesis. With this we have completed another important topic, gametogenesis. As I have been telling you, after the lecture you make notes, do the assignment questions which I will be posting for you. So go ahead, good luck.